Hello, everybody. This is Julian speaking. I hope you can hear me, and we will wait for another 2 minutes before we start the presentation, since there's still people joining the webinar. All right, I think we are now good to get started. So welcome everybody to this webinar. My name is Julian. I'm the CSO of SIGHT, and I also have with me some colleagues that help me later with the Q&A session. I'm really excited to host the second webinar of our Selling SIGHT webinar series. Last week, we talked about setting up efficient cell line development workflows. This is an application area SIGHT's technology is well known for. And this week, we will talk about the isolation of single bacteria from complex gut samples and other applications that can be addressed with our instruments for the isolation of small cells. So thank you very much for joining. The presentation will last about 20 to 25 minutes, and we will then have some time to address your questions afterwards. For those of you who use the GoToWebinar plugin the first time, you can expand to access more control options by clicking on this arrow here. Please note that everybody in the audience is muted. You're, however, able to submit questions throughout the presentation via the questions text box. So please ask questions whenever they come up. We will collect them and I will address them after I went through the slides of today's webinar. And we also uploaded some additional material that you can download in the handouts section, find an app note and the brochure for our instruments. So before I start to dive into our technology and our applications, let me briefly introduce who we are, what we're doing as a company. Since last year, SIGHT is part of the Selling Corporation, a young, very fast growing company that was funded in 2016. And by now, Selling is present in over 60 countries. And we have offices all around the globe to develop new products and also support our customers. Our product portfolio covers a number of different areas within the life sciences, and we provide tools for bioprinting, liquid handling, single cell isolation and analysis, as well as for imaging biological samples. Here at SIGHT, we develop instruments for automated isolation of single living cells since 2014. All our instruments are capable of label-free isolation and come with an image-based cell detection, which allows to provide assurance for clonality to document the single cell isolation process. It all started off with the SCP, which you can see here. It was launched in 2015. Our first product, and in 2018 we launched the B.SIGHT series, which comprises 3 instruments, 2 instruments for the isolation of mammalian cells, the F-SIGHT and the C-SIGHT, and an instrument for the isolation of microbial cells, the B.SIGHT. all cells use single-use cartridges to process the samples. In fact, if you look at the biggest pharma companies, most of them are already using our technology to generate cell lines or analyze their cells. And we also provided instruments to a lot of well-known labs in academia. This slide provides an overview of the applications that are addressed with our instruments. Our technology is very established in cell line development, which typically refers to generation of mammalian cell lines for the production of therapeutic proteins, which is a highly regulated workflow. For smaller products such as plasmids, large peptides, or simpler proteins, microbial expression systems are often the right choice. The workflow is somewhat similar to the development of mammalian expression systems, and today we will discuss how our single-cell dispensing technology can be also used to efficiently generate microbial expression strains. Another very exciting application is the cultivation of single bacteria cells obtained from mixed microbial samples. Here, cells are often present in a highly complex microbial community, and the challenge here is to derive pure cultures that can be further identified, for example, to study new species from gut microbiota. This is the topic of today's webinar. And the third application is single-cell analysis, and in particular single-cell genomics and transcriptomics, where we now offer a full platform solution for plate-based assays. And next week there will be a webinar where we talk about setting up a high-throughput single-cell RNA-seq and NGS library prep workflow. So what can you expect from today's webinar? Let me briefly go through the agenda. First, I will spend a few minutes to explain how our single-cell dispensing technology works for those of you who haven't got in touch with it. Then I will show you how one can use the technology to isolate microorganisms in a label-free manner. And finally, we will discuss a high-throughput workflow for the isolation and cultivation of bacteria from gut microbiota. The device that you see here is one of the instruments out of this B.SIGHT series. The instruments are all benchtop instruments that allow automated single-cell dispensing into well plates or other flat substrates. If we zoom into the printhead here, you can see the nozzle of one of our disposable dispensing cartridges, and we use an optical detection system which is somewhat similar to a bright field microscope to detect the cells inside the nozzle of our dispensing cartridges. And only those droplets that contain a cell with the right size and the right morphology is directed to the target substrate. You can also see one of our cartridges here. Before you start to isolate your cell with an instrument, the sample is simply pipetted into the reservoir of the cartridge, which can hold between 5 and 80 microliter. So you can also work with very limited amounts of sample. I will now show you a brief video of the device in action. Let me start this. In this case, it's the C side, but the process is always the same. You start the instrument, you open up the lid, you pipette your cell sample into a new cartridge, attach it to the instrument with 2 screws. We offer a agitation system that can be used to make sure the cells are kept in suspension so they don't sediment in the reservoir, which is especially useful for mammalian cells. You then insert your well plate, which is typically filled with growth medium or a lysis buffer, and then you start the run after setting up your experiment. And then the dispenser really goes from well to well and dispenses a single droplet with a single cell into every well. And after a few minutes, the instrument is done with the job. So we'll now go back to the slides. I will take a few minutes to show you how our dispensing technology works. So here you can see a schematic. This is the side view of one of our dispensing chips, which consists of a microstructured silicon part and a glass cover. And inside we have our suspension with cells. This here is an inlet, and here you have the nozzle where the droplets are generated. We also have a piston that's driven by a piezoelectric actuator and our microscopic camera system. During droplet ejection, the piston moves inwards and thereby displaces the liquid up and down, and this generates a droplet on the nozzle, which eventually breaks up. And when the piston retracts, new liquid is drawn into the chip. The meniscus here at the nozzle remains stable due to surface tension. It's a very small nozzle. And then we are ready for the next dispensing step. In contrast to flow cytometry, where droplet generation is based on continuous breakup, we use a drop-on-demand dispenser that is similar to the principle used in an inkjet printer. This results in very low shear forces to not impact the cell viability and integrity, and enables us to image and sort the cells. We will now switch the view. Here you can see the front view of our dispensing cartridge with some cells inside and a small pneumatic shutter that allows us to remove droplets after they are ejected. Using our optical detection system and automated image processing, the area around the nozzle is continuously monitored and the number of cells and other objects inside the nozzle is counted. In case there is no cell present in the nozzle, the droplets are removed directly after they have been generated, and only if there's a single cell in the nozzle, the droplets are dispensed to the target substrate with the pneumatic shutter switched off. And in case there are multiple cells in the nozzle or cells that do not match the specified properties, the droplets are also removed. Let me show you another video that provides some insights on how the software of our B.SIGHT instruments look like. In this case, it will show setting up and starting a run with the C side and mammalian cells, since it's quite difficult to show an example of bacterial cells. They're quite small and hard to see. So it starts with selecting the target substrate. In this case, it's 96 well plate. You can enter an ID and also a description for your experiment and set up your well plate and lay out how many cells you want to dispense into which well. The dot plot here helps you selecting the right gating parameters, which in this case is the cell size and roundness. We'll have a few wells where we dispense multiple cells as a control, and then we are ready to start the experiment. The software will then let us confirm that we set everything up correctly, and then the single cell isolation process starts. And you can see a live view of the nozzle on the top right corner. You see the cells approaching the nozzle, and the two circles indicate the area where we detect the cells in order to dispense them into the respective well. Each time the system pauses, a cell was selected and was dispensed to a well, and you can see the progress by looking at the plate. Each time the check mark occurs, the cell was dispensed. It will take around 2 to 4 minutes to fill a 96 well plate with single cells. And this is this will be the same if you work with bacterial cells. So let me go back to the slides. In the B.SIGHT series, I already mentioned we have 3 instruments. The C-SIGHT, our instrument for label-free isolation of mammalian cells. The F-SIGHT, which comes with an additional fluorescent laser to sort based on fluorescent intensity. And the B.SIGHT, which has a higher resolution optical detection system for isolating microbial cells. For each single cell that is dispensed, a series of images is stored to the hard drive to document the cell isolation process. And these are some of the images from 3 different experiments with 3 different species, Bacillus subtilis, E. coli, and Enterococcus faecalis. And what we see here is, and I marked this with a white arrow to make it a bit more clearer, we see how the cell approaches the nozzle. The third image shows actually the image where the cell is detected in the nozzle. And then the last image is an image that is taken after the dispensing event, which shows the empty nozzle to document that the cell was indeed dispensed with the droplet. For mammalian cell line development workflows, a lot of our customers use these images to prove clonality towards the FDA or the regulatory bodies. I think this gets a bit more clearer if we zoom in. So this is again an example for the Bacillus subtilis cell, which is, you can see somewhat above the nozzle. The gray circle indicates the area where the cells are detected. And if I now move forward, the cell will enter this area, will be detected, and finally leaves the nozzle. There's a growing list of microorganisms that have been processed with the B.SIGHT, which really illustrates that the instrument is compatible with a variety of different species, ranging from Bacillus subtilis, Enterococcus, or E. coli, to anaerobic organisms, but also yeast strains. Since cell detection is entirely label-free, the B.SIGHT is a great tool to isolate organisms from highly complex samples, such as diverse microbiota or environmental samples. The instrument was applied to gut microbiota from human or mice, but also to environmental samples, such as wastewater or sediment samples, as well as to clinical samples, such as sputum. All instruments use sterilized disposable cartridges to eliminate the risk for cross-contamination, and they come in a pack of 10 and are individually packed, which is especially useful when you work with pathogenic organisms or clinical samples. And this is the only part that gets into contact with your sample during the entire process. So there's no routine cleaning procedures required, and no reusable parts that needs to be exchanged. I already mentioned that compared to flow cytometry, we use a drop-on-demand dispenser, and there's no risk for from aerosols generated by this dispenser, in contrast to methods that rely on continuous droplet breakup. Given the small footprint of the instruments, they can be easily placed into regular flow benches. There are no tubings or fluids that need to be connected or deoxygenized. This makes the integration into anaerobic workflows really straightforward, as you can see on this image here. Before I will dive into the anaerobic cultivation workflow for microbiome research, I will briefly touch upon 2 other applications that can be addressed with the B.SIGHT: microbial expression systems and single-cell genomics. Cloning of bacteria and yeast is somewhat a standard method in the repertoire of every micro or molecular biologist. It's very, very common to do that. And there's a lot of applications where cloning is required, also in a high throughput manner, for example, in synthetic biology, in antibody fragment screening, or for the expression of small proteins such as antigens. And generation of such expression systems usually starts with a microbial host, such as E. coli, Bacillus subtilis, or a yeast strain. And after introducing a plasmid with the target sequence or using another method, such as CRISPR, the cells need to be cloned. Today, this is typically done by streaking the cells on agar. After streaking, the cells need to be cultured on agar for a while, followed by colony picking to bring the cells back into liquid culture. And finally, these plates can be then screened for cultures and strains that stably express the target molecule. This traditional workflow requires several manual steps, and the B.SIGHT can be utilized to directly clone single bacteria or yeast cells or other small microorganisms into liquid culture, thereby bypassing the additional cultivation on agar. And this is not only faster and more automated, it also ensures that the cultures are indeed generated from single cells. Another exciting application is single-cell sequencing of bacteria. The B.SIGHT allows for precise deposition of single bacteria into 96 or 384 well PCR plates. Using low volume liquid handling, it allows to then lyse individual cells in sub-microliter volumes for subsequent genome amplification and other assays. By combining the B.SIGHT with the I.DOT low volume dispenser, we provide a very flexible solution for plate-based single-cell genomics and NGS library preparation workflows. The I.DOT is really a great tool and allows rapid and highly multiplex dispensing of reagents for example, for cell lysis, genome amplification, barcoding, resulting in an entirely contact-free workflow. After single-cell whole genome amplification, individual cells can be then sequenced one by one. On the right-hand side, you can see a phylogenetic tree that we reconstructed from 16S rRNA sequences obtained from single cells isolated from a sputum sample. Now, in the second part of this webinar, I want to guide you through a high-throughput cultivation workflow for the isolation of anaerobic gut bacteria that was established in the group of Thomas Clavel in the University Clinic of Aachen. Here we see an overview of the entire workflow that was established to isolate and culture anaerobic bacteria. And here the focus really is on gut microbiota. And it starts with this very complex microbiome sample that first needs to be brought into liquid culture. Everything needs to be performed under anaerobic conditions to make sure the diversity is maintained and cells do not die. The samples are then pre-cultured for up to 24 hours, or they are directly processed with the B.SIGHT. And after single cell isolation, the samples were typically incubated for up to 7 days, anything between 1 to 7 days at 37 degrees in liquid culture or on agar. A first round of identification is then performed with multi-mass spectrometry. And the motivation here really is to isolate, culture, and study so far uncultured bacteria species from these samples. Such strains can be then used to either assemble synthetic microbiota. They might serve as potential new candidates for live therapeutics or provide new bacterial functions that can be used for other applications in biotechnology. To see whether single bacteria can be isolated and cultured under anaerobic conditions, E. coli and Bacillus vulgatus, a strict anaerobe, were dispensed into 20 96 well plates using the B.SIGHT, pre-filled with liquid culture medium. And the graph here shows basically the results from OD 600 absorbance measurement that was used to monitor whether the cells, whether growth could be seen in the wells or not. And as you can see, a high fraction of the wells addressed with single cells resulted in growth, 79% for E. coli and 68% for Bacillus vulgatus. Next, 2 complex samples were extracted from mice and processed using the workflow I just outlined on the slide, on the previous slide. For each sample, cells were dispensed both into liquid medium, but also some of the cells were dispensed onto solid agar filled into 96 well plates. In total, 25 plates were processed, resulting in over 1,600 wells with growing colonies after 7 days of cultivation. And as you can see here, more than 80% of the wells addressed with single cells resulted in growing colonies, and this was really encouraging. Out of these, around 1,000 samples were used for identification by multi-mass spectrometry so far. The resulting bacterial compositions can be seen in the pie charts here on the right. Note that both samples came from enriched cultures, which were cultured for up to 24 hours before the cells were isolated, which result in a slightly different relative abundance of individual species compared to the community present in the mice. The group now applies this workflow for the analysis of a variety of different samples. And for those species that do not match their multi-database, they sequence the 16S RNA for further identification. This already has resulted in several isolates of bacterial species that have not been cultured before. This is one example of such species, three previously uncultured strains isolated from either mouse or human microbiota. And this is very, very recent data, which has been presented during the DGHM conference in Leipzig around 8 weeks ago by Friesel and Professor Thomas Clabel from the University Clinic of Aachen. And I believe this is super exciting, since such culture-based workflows provide a great tool of finding and describing so far unknown or uncultured species in great detail, and actually being able to use these cells for biotechnological or clinical applications. And it's really important to understand here that more than 95 to 99% of all prokaryotic species have not been cultured so far. This so far uncultured majority provides a huge pool for potential drug candidates or other bacterial functions that can be actually used. We also have an application note describing this workflow, which you can download here in the webinar under the handout section, and also from our website. I'm now almost at the end of today's presentation. So let me briefly summarize the key aspects of the B.SIGHT, our instruments for label-free isolation of small cells such as yeast, bacteria, or archaea. It's a benchtop instrument that allows to dispense individual cells from a suspension into well plates or other flat substrates. It can detect cells between around 1 micrometer to 10 micrometer in size. For the B.SIGHT, we used cartridges with a nodal size of 20 by 20 micrometer. In contrast, for mammalian cells, with our other instruments, we use cartridges with a larger nodal size of 40 micrometer. So if you have a larger object or larger cells, you would want to work with those. The cell detection is based on bright field optics, so there's no label needed. And you can expect to fill the 96 well plate in around 3 to 5 minutes, and the 384 well plate in around 10 to 20 minutes. Due to the small footprint, it can be readily integrated into anaerobic workflows, and we also provide an automation API that allows to integrate the system with, for example, plate handling robots or other automation equipment. So how can we come together? If you're interested in our technologies or in the workflows, please get in touch with us. We are happy to arrange individual telecons. Typically, we would then proceed with an on-site demo to introduce you to the instrument and allow you to work with your cells and study the efficiency, growth, or the efficiency of your molecular workflow or assay. For now, we also offer online live demos, since travel is quite restricted at the moment in many parts of the world. So if you're interested, now it's really a chance to get an application-specific live demo, even on a short notice, that you can watch from anywhere. So please get in contact with us. Our team is happy and ready to prep a demo for you or discuss your application in more detail. We're now at the end of today's presentation. This was the second event of our series in April, of our CYTENA Selling webinar series. Next week, we will have another webinar where we address single-cell RNA sequencing. So if you're interested in these kind of workflows and in liquid handling, then please sign up or forward this to your colleagues. With this, I'd like to thank you for joining. I hope you enjoyed the presentation, and I'm now ready to take your questions. So let me see. So there's a question regarding the minimum maximum concentrations of the sample of the bacterial cell that can be loaded into the cartridge. So in terms of volume, the cartridge holds anything between 5 and 80 microliters. So you don't need a lot of sample to isolate cells. There's an optimal concentration at which we reach highest throughput and highest single cell efficiencies, and this is around 1 to 2 million cells per milliliter. This is sometimes a bit tricky to determine with bacteria or other microbial samples. So for example, for E. coli, we would typically dilute to an OD of 0.001, which is typically 100 times dilution of a day culture. And this might differ slightly for other samples, but this can be easily adjusted. Once you have the sample in your cartridge, you will see whether the concentration fits, and otherwise it's simple to adjust this. Have you isolated bacteria from uncultured samples? Yes, I mean, the workflow I presented today is just one example, and I showed you that there was already 3 species that have been identified and sequenced that were previously not been cultured. And I believe there's way more already. I think it's kind of in the process of being analyzed. And also when we performed here in-house the 16S rRNA sequencing, we detected quite a lot of cells that are that can be found in sequencing databases but haven't been cultured so far. There's again a question regarding the cell concentration, whether it's possible to dilute the cells once they have been pipetted into the cartridge. And yes, that's possible. You can simply dilute it as you would do it with an Eppendorf tube to adjust the concentration. How do you specify parameters, shape parameters for more complex samples? You might have seen this in the video of the software that I showed you. Once you have loaded your sample into the system, you can basically run it through our cartridge without actually dispensing into the wells, and the algorithm will collect a number of objects that are present in the dispensing cartridge, and they will be presented as a dot plot to you, which allows you to set up the right parameters for the cell size or roundness or others. There's another question on whether the B.SIGHT can be used to sort cells based on morphology. Yes, if the cells have a significant size difference, they can be discriminated. Say, for example, a 1 to 2 micrometer sized E. coli cell from a 3 to 5 micrometer sized cyanobacterium. This can be separated and sorted. However, for example, if you're looking into sorting rod-shaped cells from similar sized cocci, this is difficult since the 3D orientation of the cells inside the dispensing chip is somewhat random, right? So a rod can also look like a cocci depending on how it's oriented. There's a question on how we analyze the 16S rRNA from the single cells from the sputum sample. So just briefly, we performed whole genome amplification using a method called multiple displacement amplification at highly reduced reaction volumes. In fact, it was 2.5 microliter, and then followed by a PCR to amplify the 16S gene, and then we use Sanger sequencing to sequence the 16S sequence in order to identify the strains that are present in the sample. There's another question regarding the sample preparation. Somebody asked whether the fecal samples have been pre-cultured or not. And I think both is possible. The data I showed you today, I believe that the samples from mice were pre-cultured up to 24 hours before they were processed with the B.SIGHT. So let me answer a final question here. There's another question regarding sample prep. So in general, what's needed here is, it depends a bit on your sample. For pure cultures, we would typically dilute the samples with fresh PBS or fresh LB, and we recommend to sterile filter these liquids. With more complex samples, sample preparation involves typically centrifugation to get rid of larger objects, and also filtration. If I haven't addressed your question, we are happy to get in touch afterwards. Thank you again for attending the webinar, and have a great rest of the day.